One. All right, so these cells, okay, that we have inside our body are all going to be carrying out one common function, which is to keep you alive. In order for your cells to do that, okay, in order for your cells to properly keep you alive, okay, what you need to be able to do is you need to be able to supplement your cells with all the kinds of resources that you can get around. Okay, while we were discussing about this particular part, we also saw that you are going to have these different kinds of biomolecules that are going to come into play, okay, and that are going to help you in understanding how exactly you want to, maintain all of these different cellular structures. Okay, while we were discussing about these cellular structures, we also went over and we understood how exactly these cells are going to go ahead and divide and probably, carry out their different mechanisms. Now, if you look into your syllabus, okay, I'm just opening up the syllabus right here for you. Okay, so that we do not miss out on any particular part of your chapter. Okay, so here we are. Yes, while we were discussing about this, we also understood that you were going to have other molecules. Okay, these other molecules are called as enzymes, and these enzymes are going to have a very specific way in which they are going to act. Okay, it is that your each enzyme is going to have a very specific best friend, which is called as a substrate, and unless and until your enzyme gets that substrate, they are not going to be functioning well. So they definitely need their own friend in order to carry out the functions that they are specialized for. If you talk about enzymes, the only function that your enzymes are specialized to carry out is to catalyze a particular kind of reaction. Okay, this reaction is going to be to take the substrate and break it down into smaller and smaller parts. But while they are able to do that particular function, sometimes your enzymes can be tricked, and certain substrate molecules, okay, can pretend to be their best friend and make bonds with them. Okay, and that kind of bonding is called as your induced fit. Your enzymes are very, very mighty structures, but they have one flaw, which is they get denatured if they are not going to be given the exact kind of environment that they require in order to sustain themselves. Okay, now while your cells are doing all of these things, one thing that is keeping your cells in one proper position is your cell membrane, right? So you are going to have a cell membrane that is going to allow your cellular structures to remain inside the cell and not just spill over wherever the mind of the cell says so. Okay, so this is something that is very important for you to remember. And while you're talking about all of these different kinds of structures, okay, like your cell membrane keeping all of these things in one place, you also learned that the cell membrane is going to have different molecules on its surface, and these molecules are going to allow your cell to carry out certain kinds of cell communications. Okay, now some of these types of cell communication were where the cell was able to identify the self and the non-self structures. Another thing what your cell was able to do with these communicator molecules on top of it is that it was able to send a certain number of signals to other cells telling that, oh, okay, you know what, here I am, or look at that cell doesn't look really good. Or it was able to send signals to the body saying that, okay, these cells are a part of one organ system because they had different markers on them, and the other cells of the different organ system had different kinds of markers on them. All of this was essentially happening because these molecules existed on the surface of your cell. Okay, now while all of this is happening, we also learned that certain cells are going to undergo this programmed cell death. Okay, wherein if a molecule that is present on the surface of the cell gives an information to your cell saying that, oh, you know what, this particular kind of molecule that is present on your cell seems to be really a problematic structure, and maybe we should not undertake this cell with all the kinds of processes that we are doing. what you are going to be seeing is that this particular cell is going to be killed. It is going to be killed by the organelle that it so nicely curated and kept inside its own body. This organelle is called as lysosome. Okay, so this lysosome is going to come into picture, and all that this lysosome is going to do is it is going to just sort of open up, and all of the different kinds of materials that are present inside your cell are simply going to be broken down. This is called as programmed cell death. Each and every cell has a specific amount of duration for which it can survive. For example, your cells of your gut, okay, are going to replace every few weeks, and over a period of 4 or 5 years, the entire lining of your gut is going to be replaced. So if you see a person, okay, after let's say a period of 6 years, every cell in their body has ultimately been replaced. They're already a new person, okay, not just like behavior wise, or just like what they have been up to with their life, but even internally on a cellular level, every single cell in their body has been replaced by a new cell. Okay, so this is how every singular cell is ultimately going to get under the knife, okay, and every single cell is going to get killed in the body of an organism. Now, if that is true, okay, and we say that every singular cell is going to have a certain amount of lifespan, and every cell is bound to die at some point of time, you should also have a process where newer cells are going to be formed. And this is what we are going to talk about today: how exactly new cells are going to be made in the organism's body, and what are the different mechanisms in which your cellular representation can be kept similar, so that you do not like one day weigh 40 kgs and next day weigh about like 800 grams. You don't want that to happen just because your cells underwent a process called a cell death. Okay, so we are just going to go ahead and explore how the cells are going to maintain their numbers, and what exactly is the process that they are going to undertake in order to divide, and in order to just stay the way they are. Okay, so in this particular chapter, what you are going to be learning is something called as your cell division. Okay, there are two major ways in which the process of cell division takes place. You have explored these parts even before, okay, in your previous grades. You have learned that there is something called as meiosis, and there is something called as mitosis. Okay, you are going to be seeing that all of these different processes like your meiosis and mitosis are ultimately going to allow your plant cell or an animal cell to divide in number and grow, and also undertake the process of reproduction. Okay, so let's just start sharing our screen, and let's see what happens whenever you are going to have the process of meiosis and mitosis take place, right? So as soon as my screen becomes visible, we will start with that. Right here now we have the screen that is going to talk about all of these processes. Now, the first thing, okay, is where we want to explore why did this particular chapter, or why did this particular concept even come into being. Okay, so over a period of 1,000 years ago, okay, people were very inquisitive about finding out what happens when a person dies. Okay, and why exactly does a person have to die? They couldn't just agree to the natural flow of life. Okay, your human being is born, they grow old, and they die. Did not really make sense, because there was absolutely no reason why an organism just couldn't keep on surviving for years and years. Okay, and this is where a lot of scientific studies started to be taken up by your scientists. Okay, and they were just trying to figure out why exactly can't you just have your organisms undertake the process of just growing old and old and old and older, and finally just live as an old person. So there was a lot of studies done, and we figured that the reason why your cells, okay, ultimately die, and therefore an organism, is because there is a cellular machinery that stops functioning. Okay, today we are going to explore that cellular functionary, okay, that cellular machinery, and we are going to learn that how exactly we could, okay, maybe sometime in future stop the organisms from dying. That sounds really, really amazing, but that is also very, very costly affair. Okay, so you don't want to just like hypothesize that maybe in the next 20 years humans are going to become immortal and things are going to be different. It's not going to be that easy and it's not going to be that quick. Okay, so let's talk about the first mechanism that your cells are undergoing or are going to undertake, which is called as the process of mitosis. This is a very common cell division that takes place not only in the higher organisms, but even in the most lower kind of organisms. Okay, so I'm just gonna write over here. We have something called as mitosis. So I'm gonna write mitosis. Okay, this is a very, very simple kind of cell division where you have one cell that is going to go ahead and divide and form two cells. Now, this is how they taught you when you were in very, very primary grades. Okay, like for example, 8th or 9th standard. That is definitely not how easy the process of mitosis is. Okay, so what we are going to do today is we are going to look into the details of your mitosis, and we are going to understand how exactly does one small cell, okay, something like a cell of your skin, is going to undertake the process of cell division, and how exactly is the cell going to form two identical daughter cells. So let's just look at this process very, very carefully. Okay, now I have this one singular cell over here. Okay, we know that at the center of the cell you are going to have a structure called as nucleus. Okay, now this nucleus is going to contain, sorry, okay, this nucleus is going to contain a certain amount of genetic material. Okay, this genetic material is going to be just simply dispersed throughout your cell, and it is just going to carry out the different functions. Like for example, it is going to make proteins. Your genetic material is going to undertake the process of deciding what functions your cell is going to undertake. Your genetic material is also going to be present over there, so it could make certain amount of proteins and enzymes and all that falana dimkana that you spoke about. Your DNA is all going to happen here, but there is one more important process that your DNA has to undertake, which is decide when it is time for your cell to undergo the process of cell division. And once your cell has determined that, okay, now is the time you want to undertake the process of cell division, the first thing that is going to happen is the cell membrane, okay, the porous nuclear envelope that you have is going to start disintegrating. Okay, so this entire nucleus is going to start breaking down like this into its cellular matrix, and then it is going to disappear. Okay, now once your cellular nucleus, okay, this nuclear membrane is going to disappear, all of the contents of your DNA, okay, all of this nuclear content is going to start condensing down. So I'm going to write what is going to happen at each and every step. Okay, so this particular step where your DNA, okay, is getting condensed and your nuclear membrane is starting to disintegrate away. Okay, this first phase of your cell division is called as an interphase. Okay, what is this phase called as? This is called as interphase. What are the key events that are taking place during this process? The first thing is, okay, your entire DNA that is present over here, okay, is initially going to be unbound like this. Okay, initially you are going to uncondense this DNA. Okay, once this DNA is uncondensed, you are going to make smaller and smaller pieces of this uncondensed DNA like this. these smaller pieces of your DNA are going to get replicated. So there is one piece of DNA here that is going to make two copies. There is one piece of DNA here that is going to be duplicated. Okay, so now where you had one set of information, that set of information has been copied and duplicated. Okay, which means that if something like this existed before, okay, where you had your information in the form of one singular chromatin, okay, now you have the duplicated part like this, and it looks something like this. Okay, so you have the before and after picture over here. This is something that is taking place while the interphase is taking place. Okay, is that making sense? Are you following what I'm talking about?
Yeah.
Yeah. Okay, now once you have this DNA that is going to get replicated, okay, this particular smaller or you can say, like, you know, a subphase of your interphase where your DNA is getting replicated is also called as the S phase of your cellular division. Okay, in this S phase you are going to make sister chromatids. Sister chromatids, right? Now, once you have undertaken this process of interphase, okay, what you are going to do is during this period, you're also going to see that these sister chromatids are going to have all of their information very, very nicely sorted, and your alleles are going to align very, very well. What are alleles? Alleles are the similar kind of genetic information containing strands that are going to be present as two variants. Sometimes the alleles could be homozygous alleles, and sometimes you can have heterozygous alleles, which means you could have an allele for tallness written either like this, okay, where both the alleles are for tallness, or you could have one allele for tallness and one for dwarfness. Okay, something similar to this you've studied when you were doing the chapter inheritance in your 10th standard. Okay, we are going to discuss that also in a bit, but right now I just need you to remember this part over here. Okay, now once you understand that this is what your cell is undertaking, once it has decided to undergo the process of cell division, okay, during this entire process your cell also undertakes two other phases. Okay, one which is called as a G1 phase and other which is called as a G2 phase. Okay, G1 and G2 both are nothing but your growth phases. Okay, so during this entire period, what your cell is doing is your cell is undergoing a process where all of the genetic material is simply getting divided, and your entire genetic material is simply growing in length and size, and this is where your cell is actually preparing to undergo the process of mitosis. Okay, mitosis is a very, very long process, and interphase is just the process where it has decided to undergo the process of cell division. The actual mitosis is going to begin only when your cell is completely ready with all the organelles and all the genetic material. So you can say that during your interphase, when you have your G phase, all the organelles are basically just dividing and growing, and they're undertaking all the process, okay, so that the cell as an entire individual is healthy and is growing all by itself. Then you come to the S phase. Okay, so I'm going to write it this way. So you're going to have G1 phase where the cell is growing, reaching its full maximum potential. Then it comes to your S phase where this where like the sister chromatids are going to be made, and you're going to have two sets of genetical copies of everything. Once this happens, you're going to grow all the organelles for the new cell. Okay, so now you have a certain number of mitochondria, but new cell would also need new mitochondria. So you're going to grow those as well, a small amount of those. Okay, everything is then going to go to your G2 phase. And once your cell has successfully completed this G phase, okay, you are going to see that your G phase is not sometimes referred to as the growth phase. Sometimes it's just referred to as gap. Okay, so you can think of it in both the senses. It's totally fine as to how you want to refer to it, but your cell is going to grow in size and organelles are going to be duplicated in your G1 and G2. Okay, if I was going to make a representation of this for you, okay, in the sense of how the cell cycle is going to be, so I'll just make this very tiny part over here. Okay, so you are going to have your mitosis take place probably somewhere here. Okay, this is all that your mitosis is going to be, starting from here to here. Okay, so let's just put a color this way. Okay, so this entire green phase that you see, okay, like this, yeah, this is going to be your G1 phase. Okay, then you are going to have a very small space like this, okay, where you have your S phase going on. Once you look at this S phase, okay, the last part that is remaining, which is your purple color over here, okay, this is going to be your G2. Okay, now all this while you are still not having two cells with you. Okay, all that you have, even while all of this is taking place, is just one cell that is really bulged up. It is at this point of time here, okay, a very small duration, like here to here, this is where your cell is going to divide. Okay, and after division your cells are again going to undergo this, come here, and then you're going to have two new cells formed. Okay, so this is what you want to remember. Okay, now once
What is the yellow part?
this yellow part is where the mitosis is taking place. This is where the cell is actually going through all the four phases called as the prophase, metaphase, anaphase, and telophase. Okay, yellow part is where the cell is actually prepping all the material to divide, and then at this part, okay, here the cytokinesis is where the cell is going to actually divide. Okay, now what we were seeing till this part was your G1 S and your G2 phase. Okay, now let's go and talk about the different phases of your mitosis. This is the entire prep of the cell to undergo the process of mitosis. Now we talk about just that yellow part, which is for your mitosis. I guess I'm just gonna save this part, and we're going to put this in yellow color so that whenever you start to learn about this, you know that this is the part for your mitosis. Okay, now your mitosis is going to be divided into four different sections. Okay, so we are going to put these four different sections here for us. We're going to talk about them in detail. So I have this one here. Okay, I'll take another one here. Okay, we'll take one more here. All of these are going to be interconnected, and you definitely want to remember how they all align with each other. Okay, very, very important that you understand that all these phases are ultimately going to be responsible for your one singular cell to undergo the process of formation of two cells. Okay, so let's just talk about the first phase, which is called as the prophase. Too big. Okay, so let's talk about the first phase, which is called as the prophase. What I like to do in order to remember this is think of the P as preparatory phase. Okay, so you are basically going to prep yourself here to undergo the process of cell division. Now, I hope you remember I told you that that nuclear material had started to disintegrate while your cell is entering the process of prophase. Okay, the nuclear membrane has completed its cell, its particular disappearance act. Okay, so now you are going to have all of those cystic chromatids present over here, and your nuclear membrane has definitely gone away. Okay, at this particular point of time you are going to see formation and more prominent, you can say, appearance of these two tiny structures in your cell. Okay, these are called as your centrosomes. Centrosomes. Okay, these centrosomes are very, very slowly going to move towards the poles, that is here and right opposite to each other. Okay, these centrosomes are going to play a very important role in the process of cell division. Now what you have in your cell, okay, is a dissolved, I'm going to write over here, dissolved nuclear membrane. You're going to have centrosomes and you're going to have these sister chromatids. Okay, now this is what you have on your hands right now. At this particular point, okay, in order to make your sister chromosomes or the sister chromatids, your DNA has undergone a process which is called as supercoiling. Okay, it is through this process that such a long string of such a long string of DNA is able to condense down to make a small chromosome like this. Okay, your nuclear membrane has definitely dissolved, and during the particular amount of during this particular event called as prophase, what else you're going to be seeing is that these centrosomes are going to start spinning something which is called as a spindle fiber. Okay, they're going to start making something like this, which is called as a spindle fiber. What the spindle fiber is going to do, it is going to function like a zip line, okay, for your chromosomes to sort of move from one side to the other. So they're going to be like zoom and go from one side to the other. Okay, so you're going to have formation of spindle fibers by your centrosomes. Okay, these are all the events that are going to take place during your prophase. This is the preparatory phase for your mitosis. Okay, during this time you have twice the amount of genetic material in you. Okay, so this is where you are still having a diploid cell. There is entire amount of information. In fact, you're going to have twice the amount of genetic material than the normal cell. Okay, this is going to be for your P phase or the prophase. Okay, now let's go to the next phase, which is called as the metaphase. So I'm going to write over here, you have something called as metaphase. Okay, again, think of your M for middle. Okay, so P is for prep, M is for middle. Here also the number of your chromosomes is going to be twice as that of a normal cell. Okay, now if I draw the cell over here this way, okay, I can definitely see my chromosomes lining up in the middle of my cell. Okay, in order to make them a little bit more distinguishable to you, what I'm going to do is I'm going to make two chromosomes with different colors, so that when I talk about them separating, you know exactly what I am talking about. Okay, so I am going to have one purple here and one purple here. Okay, along with that, what we have are two centrosomes at two ends, and what they have started to do is they've started to connect your chromosomes, okay, like this, with those spindle fibers. But your entire chromosome material is aligned at the middle. Okay, what you see here, now we're again going to name only the part that is new in the structure. This is your spindle fiber. Okay, let's talk about the events that are taking place over here. Centrosomes are going to make your spindle fibers that are going to attach to each and every chromosome. Very, very important where they are going to attach. Okay, so for every single chromosome, they're going to attach to the small part of your chromosome, okay, which is called as your centromere. Remember, very, very confusing terminologies used over here. You're going to have chromatids, you're going to have centrosomes. Okay, chromatids are going to have, so I'm going to make this thing over here. Okay, each chromatid is going to have this central part over here. This is called as centromere. Okay, mere stands for unit. So the unit of your chromosome that is at the center is called as centromere. Telo is for the terms, okay, or the terminals like this, and the unit at the end is called as telomere. So you have something called as centromere and you have something called as telomere. We are going to discuss this telomere in a small paragraph later. You just want to remember this part. Okay, so now each one of the spindle fiber is going to go and attach to the centromere of every single chromosome. These spindle fibers over a period are going to contract and they are going to pull your centromeres, okay, and the centrosomes, chromosomes toward each other. Okay, they're going to pull the centromeres and therefore the chromosomes are also going to retract toward the spindle fibers. Okay, when this is happening, okay, during your metaphase, all of your chromosomes are aligned in the middle of your cell. So we are just going to write those key events down over here. Centromeres attached by to spindle fibers. Okay, that is your first thing. You want to remember that your chromosomes, okay, are going to be aligned in middle. Okay, and another thing that you want to see towards see in this particular event is that your central spindle fiber is going to pull the chromosomes. Just give me a second, I'm just going to remove this part. Okay, so what your spindle fibers are going to do is your spindle fibers are going to pull the chromosomes toward themselves. Okay, so this is something that is going to occur when your cells are undergoing the process of metaphase. Okay, this is all the events that you want to remember whenever you talk about prophase and metaphase. Now let's go over to the next key event that is going to take place. Okay, now this next key event is called as anaphase. Okay, so I'm going to write this over here. This is called as anaphase. Remember the A for away. Okay, so the P in the prophase is for prep, M in the metaphase is for middle, A in the anaphase is for away. Okay, now pay very, very close attention. This gets a little tricky here. I'm gonna have my centrosomes. Okay, my centrosomes are going to have their own spindle fibers. These spindle fibers are pulling the structures toward them. Okay, while they are pulling these structures toward them, now look what has happened. Okay, very, very careful attention toward what has happened. So 1, 2, 3, 4, 5, 6, and 7, 8. Do you see that your X, that was like this has been split and turned into individual structures like this. Okay, this is the key event that is taking place at your anaphase. Your chromosomes are gonna get pulled apart. Okay, middle, they're gonna align at the middle just like this, and now slowly they're gonna separate out at your anaphase. Okay, so now what has happened, we're just gonna write down the key events. Your spindle fiber is contracting. While your spindle fiber is contracting, it pulls the sister chromatids. Okay, pulls the sister chromatids apart. Okay, once your sister chromatids have been pulled apart, okay, your sister chromatids are now separate chromosomes. These are now separate chromosomes. They have the same set of information. Okay, so like this hand has the same information as that hand. It's just that now these two are different hands altogether. Okay, this is something that happens at your anaphase. And then we have the next and important phase that is going to occur. Okay, this next phase is called as the telophase. Telophase. Okay, think of your telophase ka T or 2. Okay, I'll tell you why. Now it is during this particular part that your cell is going to prep for the process of cytokinesis. That is, your cell is now going to prep for the process of formation of 2 new cells. Okay, it is during this particular phase that the nuclear envelope is starting to regenerate back. You are going to have the sister chromatids that are now separated and called as individual chromosomes. Okay, they are present in 2 different locations like this. Okay, while they are present in 2 different locations, each one of the centrosome is divided and goes to 2 individual ends. Okay, once this takes place, okay, you're going to see that your chromosomes are again going to form into a decondensed form. That is, they're going to go away from their chromosomal structure and they're going to start to form that DNA fiber. Once that has happened, your nuclear membrane is going to come around your DNA and the process of your cytokinesis has begun. Okay, so I'm going to write this over here. Chromosomes. Okay, chromosomes are going to decondense. Okay, this is the first key step. The second key step is that nuclear membrane reappears. Okay, once you see that your nuclear membrane has reappeared, cell is undergoing cytokinesis. What is cytokinesis? Cytokinesis is basically nothing but that movement of your cellular material where a invagination of the membranes is going to take place, and this invagination of the membranes like this is going to result in the formation of 2 individual cells. All these steps together are going to constitute to your cellular division in the form of mitosis. Let us look at your cytokinesis one more time. Okay, I hope it has not gone away. It has. Just let us import that image back. I don't think I can get that image back. I'm just going to try to retrieve it later. Okay, so now at this particular point, you had something like this take place. Okay, this thing is going to come back. Okay, and what you are going to observe is that you have these 2 new cells that are going to be formed. Your centrosomes are also going to be duplicated. There are again going to be two centrosomes present. Your DNA is going to be now encapsulated in your nuclear material, okay, which is also like your nuclear membrane. You're going to have your chromosomes present for some time, which are then later going to be decondensed completely, and you are going to have your genetic material present like this. And there you go. You have undertaken the process of cytokinesis completely. Okay, this process has completed. And now this cell will undergo the process of growth. Okay, it will undertake the entire G1 phase. Then it will go to S phase. It will go to G2 phase. Then it will undertake the entire mitotic phases. Okay, so you are going to have, after interphase, you're going to have metaphase, you're going to have prophase, metaphase, anaphase, and telophase. Once those four steps are done, your cell will undergo the process of formation of two new cells, which is called as cytokinesis, and that is where your mitotic cell division is going to stop. It is at this particular point that you are going to have two identical sister cells that are going to be made. Okay, and these two identical sister cells, okay, that are present over here are going to be able to do a lot of things for you. Okay, you might want to remember all the important things that your interface is able to do. Okay, so I'm just gonna remove this part and I'm gonna tell you that in order to remember all the beneficial things that come out of interface, you want to remember a very simple mnemonic, which is called as doctor. Okay, undertaking the process of interphase and therefore allowing your cells to grow. Okay, your interphase allows for DNA replication. Okay, it is during your interphase that the process of DNA replication takes place. Your organelle replication also takes place during this period. So if you want to grow new organelles, you want to grow more amount of DNA, it is going to happen at the point of interphase. You are also going to see that it is during your interphase that your cell is also undergoing cell growth. Okay, it is also during this particular time that the process of transcription, okay, and the process of translation, something that we are going to discuss in your chapter for nucleic acids, okay, is taking place at interphase. You definitely want to remember this sentence more importantly. Okay, so these two processes that we are going to talk about when we discuss your nuclear or nucleic acids, okay, the entire process of replication of DNA and reading of DNA is taking place at your interphase. It is also during your interphase that your cell is obtaining all the nutrients in order to survive. Okay, and these nutrients are going to be used by the cell during its like the cell division process, during it is undertaking this mitotic growth, or it is also called as the M phase. So entire phase of your cell, okay, if I just do this, if this is where your cell is dividing, this is called as interphase, and this is called as M phase or mitotic phase. Okay, now while this is happening, your cell is obtaining nutrients during your interphase, it is growing during interphase. All these events are happening only during interphase. During the M phase, the only thing that your cell is doing is dividing, nothing else. It is not even going to respire during your division phase or M phase. All the things, everything that a cell needs to do in order to survive is going to be done only during the process of interphase. Rest of the time, your cell is just going to divide. Your mitosis is going to give rise to a diploid nucleus, okay, and this diploid nucleus is going to give you a clone of the original cell. What your mitosis is able to do for you, okay, so I'm just going to write this over here. What are the benefits of undertaking the process of mitosis are told. Okay, again, another mnemonic you want to remember. These are the things for which we undertake the process of mitosis. Mitosis is going to help your body to undertake the process of tissue repair. Okay, so I'm just going to write it this way. You can repair your tissues. It allows for the organism's growth. So I'm going to write organisms growth, apostrophe s, organisms growth. Okay, this particular kind of cell division is a key process in your asexual reproduction. Okay, and along with that, every single time when you take the process of sexual reproduction, whenever the cell is going to undertake the process of sexual reproduction, what you are going to observe is that the development of the embryo, okay, so I'm going to write over here, development of embryo is going to be because your embryos are undertaking the process of mitotic cell division. If your cells were not undertaking the process of mitotic cell division, but they were doing meiosis, you would have different kinds of cells forming, not the same kind of cells. You definitely want your somatic cells to be made out of mitosis and only your gametes to be made using meiosis. Okay, this is a very, very important thing that you want to remember. What I'm going to do for this particular part, okay, is I'm going to share with you certain number of micrographs. Okay, what are micrographs? Micrographs are nothing but the pictures taken through a microscope. Okay, these micrographs are going to show you exactly how your cell looks when it is undergoing the process of cell division. So if you could just bear with me for a minute, I will show you how your cell looks under the microscope when it is dividing. Okay, so just give me a second. I am just going to share that part with you.
Okay, so I have a question.
Yes, tell me.
So under mitosis, there is interphase,
Hmm.
and under interphase, there are those 4 phases.
No. So now that's where you got a little bit confused. This is your cell cycle. This is the entire cell cycle. Okay, starting from here. Okay, so I'm just gonna draw this here so that I can probably show it to you. No, let me just do this way. Okay, this is your entire cell cycle. If you remove this part, okay, I don't know how to do this because I did not sit with a scissor with me. Okay, this entire part of your cell cycle, this entire part without the small piece, this is entire interphase. This is where your cell is growing, this is where your cell is developing, respiring, getting all the nutrients, all that falana dimkana. This is where your M phase is taking place. This is where you're undertaking prophase, metaphase, anaphase, and telophase, and cytokinesis as well. After the process of cytokinesis is done, what you are going to have on your hands are two identical cells like this. Okay, so the process of cytokinesis is going to leave you with two identical daughter cells. That makes
So
sense?
those 4 phases are under M phase.
Yes, those four phases are under your M phase, right? So now
Yeah.
this is how your cell is going to look like under your microscope. Okay, I'm just going to show you a few images. This is how your telophase is going to look like. Okay, what is happening during the telophase? If you look, there is an invagination of the membrane and all those materials are just probably just getting condensed over other sites. Okay, if you look over here, okay, this is your prophase, this is the prep phase where your nuclear membrane has completely disintegrated away, and what you're able to see is your genetic material is decondensing so that it could form into your chromosomes. The same thing that is happening over here as well. Okay, these are the different phases where you can see the chromosomes are probably getting condensed more and more so that they could actually form your chromosomal structures. At this point, you are able to see the anaphase, and you can actually make out tiny spindle fibers trying to pull those different kinds of sister chromatids away from each other. Okay, this is another picture for your prophase. This is your metaphase. Okay, they're all just going to try to align at the center, and then they're going to be separated away. This is your prophase 2. This is where you are going to see that the cell, okay, is going to undergo the process of meiosis, and you're going to have two cells that are going to have division, and they're going to form four different kinds of cells. This is your interkinesis. That's when your cell is still undertaking the process of cytokinesis. The two cells are still encapsulated within one singular membrane. Okay, and these are the different stages seen inside one singular micrograph. This is seen under your onion root tip. Okay, so if you take an onion root tip and you stain it with your eosin, this is probably something that you are going to be able to see. Okay, I'm going to share with you a few other pictures so that you are able to identify these different kinds of your stages of your cell division more easily. So I'm just gonna do this mitosis pictures. Okay, and then I will share my screen with you so that you can see that. So here you go. There are different resources and images available on your Pinterest. You can just definitely go ahead and look at that. This is something that has been drawn. Okay, if you want to look at the pictures in more detail, this is how you're going to be able to see that. So I'm just going to type some keywords here for you so that you could use that for making your own notes. So I'm going to say mitosis under microscope. Now, these are all the things that, just automatically come into my search history because I probably search them for every year. Okay, but you might have to do a little bit of more digging in order to get these pictures. So here is a very beautiful picture where you're able to see all those phases taking place. Okay, now I'm going to show you one very important picture, and I want you to try to identify what is happening in each one of those. Okay, so now can you tell me which one of these cells over here, okay, is duplicated all of its nuclear material? Which cell looks like really huge and plump and feels like, okay, there could be double the amount of genetic material? Do you think it is going to be this cell, or do you think it is going to be this cell, or do you think it is going to be this cell? Which one?
The first one.
This one? And what do you
Yeah.
think about this one then? What has happened with this guy? It does not have the double amount of genetic material, just like the normal quantity, it seems. So could it be because this cell has just divided and does not have, doesn't seem to have decided to undergo the process of cell division yet? So when you're going to see a tiny cell, okay, just something like this, you're probably going to say that the cell just underwent the process of cell division. But this cell over here is prepping to undergo cell division. These are the different phases where the cell division is already occurring. When you look at these different micrographs, okay, you want to make sure that you understand these subtle differences between them, because it is these kind of pictures, not the colored ones though, okay, it is these kind of pictures that are going to come into your exam, and you're going to be asked to identify exactly what is happening here. In order to help you learn this in more easier manner, okay, there are certain amazing videos that have been put up by different people. Okay, there is this one channel that probably does an amazing job at teaching us about this. It is going to be your amoeba sisters. I'm pretty sure I've shared a few videos about the work of these people before with you, but the mitosis video is definitely something that you want to check out. Okay, so what I'm going to do is I am simply going to share this guide, not the guide, the video with you on your WhatsApp, and I'm going to ask you to view it and then come back to me in case you have any doubt. Okay, so here you are able to see the link for your video, and do let me know if there is anything that you want to discuss or anything that you did not understand.